Behavioral Neuroscience, Chapter 3, Segment 2. Starting off with the Goldman equation. Unlike the Nernst equation, the Goldman equation predicts the differences in charge across a membrane rather than the equilibrium potential. This takes into account the concentrations of ions on either side of the membrane, along with their permeability to the membrane. It's useful in predicting the effects of ions and their concentrations on the possible membrane potential. And you have calcium ions, which are neurons use ion pumps to keep levels of calcium cations low. And then moving on to a threshold amount of depolarization triggers an action potential. Action potentials are changes in neuronal potential, which triggers an electrical message to travel through the axon into the presynaptic axonal terminals. Hyperpolarization is the continued negating of charge within a neuron, increasing the membrane potential. Depolarization is the decreasing in membrane potential, which leads to a charge difference of zero between the inside and the outside of a neuron. Local potentials are a phenomenon in which membrane responses diminish the farther they travel from the source stimulation. Also arise in synapses in response to other neurons. The threshold is the point at which a membrane potential of around negative 40 millivolts causes voltage-gated sodium channels to open, which creates the spike in charge that commences the action potential. The all or none property is the behavior of an action potential to occur at full amplitude or not to occur at all, meaning there is no middle ground. When a stimuli far exceeds a neuron's threshold, multiple action potentials occur instead of one larger action potential with the larger amplitude or even a smaller one occurring. In other words, the strength of a stimuli only affects the frequency of the action potentials, not the amplitude. After potentials occur after an action potential occurs, and the return of resting potential involves, oscillations. And these occurrences are related to the movement of ions in and out of the cell. Ionic mechanisms underlie the action potential. Alan Hodgkin and Andrew Huxley, using large squid axons involved in escape behavior, which are visible to the naked eye, studied their action potentials. They replaced their intracellular fluids with other ones to measure its effects on action potentials. Voltage-gated sodium channels are transmembrane proteins, and they are selective to carbon cations. They open once depolarization reaches threshold levels, causing the spike in charge that leads to an action potential. They must open in order to begin the action potential. It's the key to causing the threshold. Refractoriness is the behavior after an action potential occurs, where the axonal membrane becomes refractory, meaning that it becomes unresponsive to stimuli that occur immediately after the first. This occurs in two phases afterwards. There is absolute refractory phase, which is this really brief phase where it's completely unresponsive. No matter how much stimuli you apply, no action potential will proceed or come afterwards. Relative refractory phase follows the absolute refractory phase and is a reduced sensitivity during which another action potential may occur, but only with a very strong stimulus. And the axon hillock is worth noting. It's the swelling of the axon where it projects from the soma, and it's where the action potential is either released or not. Action potentials are actively propagated along the axon. If we look at box 3.1, there is voltage clamping, which is a technique which was important, an important preparatory measure used on the squid axon by Hodgkin and Huxley. It involves stimulators being used to control an axon membrane to remove at a given potential or voltage. No, don't go down there. Oh boy. What are you doing? Patch clamping is a method superior for gathering more details to voltage clamping and an individual pulls away a tiny patch of membrane covering the microelectrode, uh, and this tiny patch's currents are then monitored using a voltage clamp. Now, the conduction velocity is the speed at which action potential travels. Uh, an action potential travels across an axon, and it's dependent on the diameter of an axon. It's slower in thinner axons and faster in thicker axons, and this is because larger axons allow depolarization to travel with less resistance through the interior of the axon. Another important thing to note are the nodes of Renvia. They're gaps in the myelin sheaths that surround axons, and they improve conduction velocity because, see, the myelin sheath actually creates resistance in the travel of ionic currents. And so nodes have these sodium channels which open, allowing action potentials to leap from node to node. And this is a process known as saltatory conduction. And you can see the root saltar in there, which means leap, like somersault or and such. Then you have box 3.2, and in the beginning it discusses channelopathies, which are medical conditions involving abnormalities in the form and function of ionic channels caused by mutations of relevant genes. Then you have tetrodotoxin, which is found in the ovaries of a pufferfish. It selectively blocks voltage-gated sodium channels, which are key to action potentials. So, because no action potentials occur, this leads to paralysis and subsequently death. Then you have saxitoxin, which sounds like a weird saxophone. It does a very similar thing. It also selectively blocks voltage-gated sodium channels, which has the same effect, but it is found in a form of algae known as red tide. Where are you? Don't go there. And then you have batrachotoxin, and it's a potent neurotoxin like the ones before, that's produced by South American poison arrow frogs. But what's different is it actually forces sodium channels to remain open, which also has its own lethal effects. So synapses cause graded local changes in the postsynaptic membrane potential. So one thing to know are what neurotransmitters are, and there are these chemicals that travel across the synapse from axon to dendrites to communicate with the postsynaptic neuron. They're carried by transport vesicles to the presynaptic membrane and then into the synapse through exocytosis. So it merges with the membrane and then releases the neurotransmitters. And so these synapses can be excitatory or inhibitory, right? So postsynaptic potentials are the brief change in membrane potential of a postsynaptic cell subsequent to the release of a neurotransmitter into the synapse. Neurons receive multiple postsynaptic potentials all at the same time, and they can collectively determine whether an action potential will occur. Excitatory postsynaptic potentials, or EPSPs, are a depolarizing potential in the postsynaptic neuron caused by excitatory connections. And then each subsequent EPSP increases the chance that the postsynaptic neuron will reach the threshold and fire off an action potential. And so one last thing to consider is synaptic delay. And this is the delay in time between the arrival of the action potential to the presynaptic it's a delay in time between the arrival of the action potential to the presynaptic terminal and the postsynaptic depolarization of the postsynaptic neuron. This is actually caused by the amount of time that it takes for the release and diffusion across the synapse that a neurotransmitter must go through. No! Chill out. Okay. That is it. Signing off. Goodbye.